Alright, so everyone, welcome to our lecture 4. Lecture, not tutorial, okay. Okay, so this week is very important because for every biopsychology module, we always talk about vision. So I cannot re-emphasize how important it is. Right? It is relevant for midterm, of course, yeah, right. Okay, so just to start things off, okay, vision or sight. Yeah, so it's just some trivia, right? Some general knowledge. So vision, out of the 5 senses, is the one that we human beings, okay, and mammals are primates in general, okay, we rely heavily on vision, and it covers around 50% of our cerebral cortex. And surprisingly, we only have 3% dedicated to hearing, that is our temporal gyrus, our middle temporal gyrus. 11% for touch, right, primarily located in our central sulcus, that's our somatosensory cortex, okay. Right, so from sensation to perception, all right. So one of our first slides on psychology, right? This is, I would say, cognitive psychology, right? So this is also a revision, okay, of our intro to psychology. I just want to emphasize on the words in bold, right? So sensation is the process of receiving information, right, about the environment and how we transduce or translate such information into action potentials. So it's conversion of one energy into another. And perception is different, okay? Perception requires some kind of interpretation, some form of higher order thinking. So that's the way it differs from sensation. Right, so take note of the difference. Very, very important. Right, so here, on our this slide, the right panel, right? So we have just some, very, very colorful diagram, like a light coming in, and we're seeing its color, right? That reinforces our photoreceptors. Right, so 2 types of processes. Again, this is a revision, right, from our intro to psych. So we have bottom-up processing, and we have top-down processing. So bottom-up processing is we're really focused on sensation, right? Information from our 5 senses, right? Top-down processing is different. We are receiving information from our 5 senses, yes, but at the same time, we're making use of our past knowledge, right? Our experiences, our memory, right? Once we make use of our memory, that is top-down processing. And top-down processing also involves our prefrontal cortex, heavily involved, always involved. Right, so this is, where is Wally, right? I recall, if I'm not mistaken, where is Wally? Can you find Wally, right? I couldn't, right? So I leave it up to you guys, right? This is a very, very famous comic strip character, right? So light travels in waves, right? Revision of our secondary school physics, for those of you who still remember. So surprisingly, okay, light, after all these years of experiment, we have reached a consensus. So light can exist both as a wave, right, and as a particle. So it has dual properties. We learned this from this experiment in physics, very famous experiment. We call it the double slit experiment. Okay, you guys can Google it out. Okay, there's a lot of videos on this. Very, very important seminal physics experiment, right? So strictly speaking, I would say it, right? Me and a lot of physicists would say that light should be conceived as waves of electromagnetic radiation, EM radiation, with photon as our fundamental unit, right? So photon is really like the packets of light particle. And electromagnetic waves, right, are all around us, and they travel rapidly. I'll correct this sentence. I know it's grammatically wrong, right? So wavelength, take note of it, right? Wavelength equals to distance between the peaks of 2 waves, right? So it determines the color, right? Determines the shades of gray that we see. Amplitude is loudness, like what you're hearing me right now, okay? Through the speaker on top of the ceiling, right? So that determines the loudness of sound, that determines the brightness of light, right? Higher amplitude, the brighter the light. EM spectrum, right? On this slide, okay, we see that light from the sun, right, contains a variety of wavelengths, right? So they're involved in just 3 processes: absorption, reflection, refraction. We'll talk about them one by one. These are all revision of our secondary school science, right? So air and water refract, right? Will change the direction, change the speed as well, right, of light. So the EM spectrum, when we talk about EM spectrum applying to visible light, right, it's between 400 and 750 nanometers. Remember this, right? This is very important general knowledge. So for blue, we are talking about the short wavelengths, right? The shorter wavelengths, right, from 400 to about 450. The long wavelengths will be red, right? So that's like 700, roughly 700 to 750. Right, so when it's dusk, when the sun is setting, we are seeing a lot of red colors, right, long wavelengths. EM spectrum continued. So as said, right, range of energy visible to humans falls between 400 and 450 nanometers. The rest is not suitable for human visual system. They will damage our eyes, they damage our bodies, okay, X-rays, microwaves, radio waves, right? X-rays and microwaves are very, very invasive, right? You don't want yourself to be exposed to them. Radio waves, no, right? Radio waves, similar to Wi-Fi, long wavelength, it will not really affect our eyes, our body. So extremely short, UV waves are visible to other species. Surprise, okay? Insects and birds, and snakes can detect infrared light. Yeah, so very surprising. Yeah, it helps their survival, right? I would say that. Okay, so we have some people coming in. Okay, so light interacts with the environment through reflection, absorption, the two main processes. So on the right, we can see that this pen is red, right? Because it has these physical properties that allows it to reflect long wavelengths of visible light. Right? If it has the properties that reflect short wavelengths, we'll see it as blue, right? So color, in other words, is a kind of perception, right? It's not entirely sensation, right? We still have to do some kind of top-down processing, some kind of interpretation. Again, right, so we have this light to heat energy, right? Light can be converted into heat energy. We all know that. This is primary school science, okay? And black object absorbs all wavelengths, and then it's converted into heat. So white reflects all the wavelengths. So light is not totally converted into heat, right? So this is just a recommendation. So on a hot day, it's better to wear a white shirt, okay, like me. Okay, I'm always wearing a white shirt, yeah, than to wear a black shirt, yeah. So again, right, so we have the third process called refraction, how light interacts with the environment. So this one we all know, light is bent as it travels through water, right? And this leads to distortion in how we perceive, right, the image, right, the image of a straw, right? So this is very easily explained because when light passes through a different medium, in this case, this is water, right? Okay, a liquid. So it changes its speed of travel, right? And it's really the speed of travel that leads to the bending effect. Yeah. All right. So first quiz, very simple. Everybody know this, okay? What happens to light waves when we see a rainbow? Right, this is our tutorial as well, right? Okay. Reflection, absorption, refraction. The answer is actually in the slide itself, right? So how many of you pick A? Nobody, right? How many of you pick B? There is a kind of absorption, but it's not a perfect answer, right? C? Hands up, right? Yeah, so everybody pick C, right? Of course, it's refraction, yeah? So light is refracted into its primary colors, right? So now we come to something that's more biological, something more physical, very important. Okay, so structures and functions of the visual system, right? So on the right panel, first of all, we see a hollow cavity. So that is really the place where our eyeball is contained, right? So protecting the eye, we have a bony orbit, right? And we have lots of fatty tissues, right? And our eyelids and all this blinking, having tears, surprisingly, they do kill all bacteria. Yeah, they keep our eyes clean. So very important reflex, right, that we need, right? Okay, anatomy of the eye. So this is more like an exterior, anatomy of the eye. So we have three important structures, right? So number one, whenever we look at a person's eyes, right, the surroundings, the periphery of it is always white, right? So that's what we call the sclera, right? So if you read a lot of literature books, they call it the white of the eye. Right? You're looking at the man in the white of the eye. That is the sclera, okay? So it's a tough outer white covering, and it stretches all the way. It covers our entire, eyeball. Not just the front part, right? And then the iris is the circular muscle, right? We all know this, the circular muscle surrounding the pupil, controlling pupil opening. And different shades of color for people of different ethnicities, right? So for Asian people, generally it's brown. It is not black, okay? It is brown, okay? Just different shades of brown. There's no black iris, strictly speaking, right? Don't get this thing wrong. So pupil opening at the center of the eye, okay? The pupil is black, right? Because internally our vitreous humor, it's, that part is really, that part is dark, that part is black, yeah. Okay, we'll come to that, right? So anatomy of the eye continued, right? So the first part, right, that interacts with light is the cornea, right? So the outer transparent covering, right, protecting our eye, okay? And it bends the eye towards the retina, very important. So if it cannot do this thing properly, we get astigmatism. And I just want to mention we have this thing called aqueous chamber, vitreous chamber. So don't get the chamber confused with humor. The chamber is just a physical space. The humor is the liquid-like substance. right? So for aqueous chamber, it contains, right, the aqueous humor. And then through the pupil and the lens, we have the vitreous chamber containing the vitreous humor. And the vitreous humor is like, jelly-like substance. It's not fluid, it's jelly-like, okay? It's more solid than fluid. So lastly, we reach the retina. That one we'll talk a lot about it. The retina is so important. Right. So the retina, to begin with, we have the macula, right? It is an area of the retina that is not covered by blood vessels. Okay, take note of that. And its radius is quite big, okay, 6 mm. You can look at your ruler and see how thick 6 mm is. Right? It's quite a, at the body level, right, 6 mm is quite a lengthy, quite a length, I would say. Within the macula, we have a small pit, and that is called the fovea. Everybody knows this, okay? So fovea, we need it for highly detailed vision. Its diameter is much smaller, 1.8 mm. Okay, this is the interesting part that our author wrote in the textbook. Hopefully you guys read about this, okay? So it says that primates, including us, right, are the only mammals whose foveas contain only cone photoreceptors. So we don't really have rod in our fovea. Take note of that, right? Other mammals like cats have foveas with both, right, cone and rod. So their night vision is better, but unfortunately, because they don't really have as many cones as we do, their color vision is not as good as us. It applies to cats, applies to dogs as well. Right, importance like central versus peripheral vision. Okay, so I just want to highlight the two keywords or the two key terminologies. Right? So one is called photopic color vision. That is tied to central vision. So whenever we talk about central vision, this is the kind of vision we're talking about, highly detailed color vision. Peripheral vision, right, by contrast, is tied to scotopic vision, right, dim light or simply night vision, right. No color, very minimal color involved, right? So we need peripheral vision because, without peripheral vision, it's very hard to move around, right? We cannot perceive contrast, we cannot perceive motion, right? We cannot really perceive all the color, right? Important, right? We still need this thing to give us a sense of movement, right? Landmarks of the retina. So just now we talk about macula, we talk about fovea, right? All these things. So what is new on this slide is this thing called the optic disc. So in our tutorial, we'll talk more about this. But centrally, just take note of the location, right? So on the right panel, yeah, it is highlighted in yellow. That's the yellow spot. On the left panel, I mean. On the right panel, basically, it's somewhere just right, in front of the optic nerve. Because it is the area in which our optic nerves exit our eyeball and passes all this visual information, right, to our brain, right? So as said, right, blood vessels, axons of optic nerve, exit eye in the optic disc, and this optic disc does not, remember, does not contain photoreceptors. And because of that, it has no photoreceptors. It's called a so-called blind spot. Yeah. Not to get mixed up with driving. Okay, this is the blind spot of the retina, right? Not the blind spot when we are driving. Okay, structure of the retina. I'm going to be a bit slow on this because this part is a bit non-intuitive. I will talk a bit more in tutorials as well. Okay, so take note at the very top, when light is coming, the first thing that light falls on is, surprise, surprise, the optic nerve, the axons of the optic nerve. And this is the part in which I think improper labeling have been done. I would label it as axons of the ganglion cells, right? To be more precise. You guys can change that, okay? Axons of the ganglion cells, right? So note that it is not the photoreceptors that is closest to light. Surprisingly, they are the ganglion cells. Our photoreceptors are way below. Wow. So when you look at the diagram, they're way below. They're really tucked underneath. Really tucked underneath our photoreceptors. And then we have this thin layer of epithelium, pigmented epithelium, that does not really send out any nerve endings. Which means that our nerve endings, right, they belong to the ganglion cells. So all this energy from the cones, the rods, pardon my pronunciation, right? So they have to travel in a bottom-up direction all the way to the ganglion cells. And then the ganglion cells send out all this energy, send out all this nervous outputs to our brain. So this is a very, very non-intuitive, unintuitive biological structure. So light comes in, right, goes through all these layers, and then later on, right, our photoreceptors sends out all these outputs back to the ganglion cells. Ganglion cells package them and then sends out all the outputs to our brain. Wow, wow. So it's a loop, essentially, okay? Light comes in, bringing all the energy, top down, then bottom up from our photoreceptors, goes back to the ganglion cells. Yeah. From the ganglion cells, send out to the brain. Wow. This is incredible, right? I still find it incredible after all these years. Yeah. Okay, so now let's talk about our photoreceptors. So the first thing first, right? So we have this transduction of visual information, primary function of our photoreceptors. So conversion of all the EM energy from light into electrochemical energy, right? That the other cells like bipolar cells, horizontal cells, right, ganglion cells can process. So photoreceptors are simply named after their shape, right? So a rod literally look like a rod, a cone literally look like a cone. So we have around 90 million rods, but only 4 to 5 million cones, right? So rods, biologically speaking or evolutionary speaking, right, they are preferred over cones because, you know, peripheral vision, you know, is just so much more important for survival, right? Sensing direction, sensing motion, you know, contrast and all these things, so much more important than sensing color alone, right? So that makes sense from an evolutionary perspective. And then the last part I want everybody to take note of is that we have 3 cones, right? 3 photopigments, right? For processing the 3 primary colors, red, green, blue, right? So here this is from our textbook, responses of rods, responses of cones to different wavelengths, right? So we have 3 types of cones, right? As said, we have blue, we have green, we have red. Yeah, so just take note, right? So they're all pretty much kind of inverted quadratic in nature. All right, so further information on the differences between rods and cones, right? So cones require more light than rods in order to respond. That's understood, right? So only during daytime can we see color, right? Under bright light. Yeah, so we have 3 different cones, as said in a previous slide, right? And they're only found, they're right, high density in fovea, right? And for mammals like us, primates like us, we only have cones in the fovea. Now for rods, right, they contain this thing called rhodopsin. So it's a special kind of photopigment, right? And then it's found in high concentration in the periphery of retina. Okay, and then we have this superior vision in dim light in the blue to green range of this EM spectrum. Just take note of this, right? Okay, so quiz time. So we have a dog. Take note, this is just very important information, right? Okay, so dogs only have 2 types of cones, right? Blue and yellow, right? So consequently, our pet dogs are essentially, you know, suffer from red-green color blindness. So it's very hard for them to see a red ball on the grass, right? So they cannot see as sharp as us, right? And not so many colors. They can be as good as us when in motion and under dim light conditions. So, vision differences between humans and dogs due to cones or rods. All right? How many of you say cones? Hands up. All right. How many of you say rods? Hands up. Yeah, cones, right? Cones. Yeah. For cats and dogs, right? That's how they differ from us humans, right? Cats and dogs do not have as many cones as we do. All right, so now we come to 2 very important slides. I want everybody to take note, okay? Remember this thing. Very interesting information, right? How do rods transduce light energy into neural signals? Right, so EM energy into electrochemical energy in our retina. So take note, right? So the surprising part about a rod is that its resting potential is not negative 70 millivolt. It's negative 30 millivolt. And this resting membrane potential, to me, in my humble opinion, right, it's special because this actually facilitates depolarization. It actually facilitates the membrane potential into becoming more positive, right? And what really brings about this positivity is the dim light, okay, when it's in the dark, when it's not so bright, right? And we have this movement of sodium ions, right, that actually start this depolarization phase, right? So this movement of sodium ions is what we call the dark current. Yeah, what a name, what a name, yeah. So, but do remember, it's called a dark current, okay? Believe it or not, we still accept it, yeah. So because it's in the dark, right? And take note that all these sodium ion channels, right, they are actually on this metabotropic receptors, right, that really respond to this secondary messenger, CGMP. Okay, very, very long word, right? So it's this cGMP messenger that actually maintains this so-called dark current. And it's because of this influx of sodium ions, right, that we get this depolarization, right? So depolarization of what? Always remember there's a depolarization of the rod membrane potential, right? And consequently, we get this release of glutamate into the synapses. And it's because of the glutamate, right, that we get this higher occurrence of excitatory signaling. So because of this excitatory signaling, we can see in the dark. Yeah. So it's a chain, it's a cascade of events. Right? Take note of this. Ultimately, just remember, it's depolarization has brought about all this outflow of glutamate into the synapses. All right, so transduction in rods. What happens in rods in response to light, right? Under bright light. So at the back of our lecture hall, what is happening, right? Okay. So what happens is that we have this photopigment, right, for rods. Remember? So it's called rhodopsin. So you undergo a transformation from cis-11 to all trans. All trans is unstable, and you can easily break apart into opsin and retinol, its constituents. Right? And because of that, what we have is that we have a release of enzymes. Right? What these enzymes are called, we see in our tutorial videos. So cGMP gets broken down. Because of that, right, our ion channels close, unfortunately. So we have less glutamate into the synapses, lower excitatory signaling. Okay, it's lower excitatory signaling. Remember that. It's not higher inhibitory, but it's just lower excitatory signaling. And so, right, our rod membrane potential becomes hyperpolarized. So it becomes more negative than negative 30 millivolts. Right, it can be negative 40 millivolts. Okay, so the last bullet point is something very interesting, right? So, however, if we go back to a dark room, what will happen is that our rhodopsin will form again, right? Through a series of enzymatic reactions, by merging opsin and retinol together. Just take note of that, right? Okay, so let's carry on. Processing by retinal interneurons. Okay. So we'll talk about all these cells, right? All these so-called interneurons, one by one. So first of all, okay, just a trivia at the top, we have 95 million. Wow, that's a lot, you know. So we have these horizontal cells. That's what I bracketed in this rectangle. It's like an alien, okay? Alien octopus, all right? And form connections with photoreceptors, with bipolar cells. And it contains this graded potential. So it's either like a slight depolarization or a slight hyperpolarization. So it doesn't really lead to something like an action potential. It's just slightly more positive or slightly more negative. Yeah. So bipolar cells are much more interesting because they are the so-called, they are part of the receptive fields. So a receptive field is just something, any area, okay, on a retina, okay, that really receives the light, okay, receives the light and passes the light energy to all our retinal interneurons. Okay, basically, receptive, okay, you're receiving the light. Okay, the field is just an area on the retina. It's just that simple. So, but what I really want people to really encode, right, in your mind, your brain, right, what I put on in bold, right? So antagonistic, okay, we see this word again. Last week we talked about them, okay, and now we see it in the retina, right? So we have this antagonistic centers around organization, and it's maintained, okay? It is maintained, the word is maintained, okay, by lateral inhibition. So we have two types of bipolar cells. Okay, one is diffuse, one is midget. For the details, you guys can refer to your textbook, okay? This is not so crucial, right? And again, okay, they have graded potentials, right? So on this slide, I want everybody to remember this thing very, very well, okay? That's why I put double asterisk there. Double asterisk means it's highly, highly important, right? Okay, so first of all, so we have this on-center, off-surround structure, organization, right, for one kind of bipolar cell. But at the same time, we also have this less common off-center, on-surround, okay, organization. Less common, okay? Let's just talk about the on-center first, right? So when it's on, that means a bar of light, okay, let's just a bar of light, for instance, right, is falling on the center, okay, the center of the receptive field. And that leads to depolarization. Okay, depolarization means you're going to get more excitatory signaling, okay, more release of glutamate, for instance. And then the off-surround, okay, the light is also going to fall on the surrounding region, right, the periphery of the on-center. Okay, but it's only like its most, its closest region, right, not the entire stretch of the retina, right, the closest region. And that is going to lead to hyperpolarization, right. So less excitatory, but it can also be inhibitory response, right. Most of the time it's inhibitory in nature. Okay, so off-center, on-surround is basically the direct opposite of what I said. Okay, I'm not going to elaborate more on that, it's just the direct opposite of what I said. Right? So lateral inhibition, okay, in this case, very, very important. I'm going to read out this entire sentence, okay? So basically, it's like we have all these active photoreceptors, okay, rods, cones, and right, the horizontal cells. If you cannot remember the horizontal cells, perfectly fine, but do remember the photoreceptors, right? Because receptive fields always involve the photoreceptors. Right? So they are actually limiting the activity of the neighboring less active cells, the so-called off cells, and they're doing it in a horizontal way. So on the diagram on the right, okay, note the line in black, gray, in bold. So it's doing it horizontally. And all this, so it's the direction, the lateral inhibition is running on a parallel line, horizontal line. So yeah, so that's how it works. Okay, line by line, row by row. That's why it's called lateral inhibition. Right? So take note of that, right? Okay, so now we come to amacrine cells. So they are the cells between our bipolar and ganglion cells, right? They also have connections with other amacrine cells. But primarily, I mean, it's really for sending information from our bipolar cells back, right, to the ganglion cells. Okay, so ganglion cells, very, very interesting. They are also part of our receptive fields, and they also have this antagonistic centers around organization, right? So if the question asks you which two types of interneurons have this antagonistic, right, centers around organization, you should say it's ganglion cells and bipolar cells. Right, don't get that one wrong, right? And then we have like 3 kinds of ganglion cells, right? So we have M, we have P, we have K cells. I'll elaborate on them later, right? Okay, so we have this very, very colorful slide, colorful diagram showing the receptive fields of bipolar and ganglion cells, right? So like the bipolar cells, they also have this antagonistic centers around organization, right? This is the third time I'm saying this, right? However, right, ganglion cells, they produce action potentials, right, APs, not graded potentials, right? Graded potentials are what I said, they're just slight depolarization, slight hyperpolarization, not strong enough, right? The slight depolarization is not strong enough into becoming an action potential. So they're virtually almost all, on all the time, they're active all the time, ganglion cells. So they fire once per second, right? Even without stimulation. With stimulation, they'll spike, fire at a much faster rate. Yeah. Okay, so now everything is at the retina, at the eye level. Light goes through the lens, falls on the retina, right? And all these cells, we talk about all these retinal interneurons, right? So what happens, and then what happens? So we have to look at this, optic nerve connections, right? Neuroanatomy. Very, very important. I'll be going through this thing a little bit slower, right? So note that we have this optic chiasm, right? That's where the axons, the optic nerves, right? Optic nerve fibers from both of our eyes, they converge, right? Somewhere in front of our thalamus, right? Then later on, we have this lateral geniculate nucleus, LGN. So this is possibly the most famous nucleus whenever we talk about visual processing. Okay, so it receives around 90% of optic tract axons. I wouldn't really talk about all the layers in details, but just know that we have 6 distinct layers, right? They really keep input from each eye separate. And surprisingly, we do have the hypothalamus involved, right? Even though it's not really shown clearly on this diagram. So we need the hypothalamus to regulate our circadian rhythms. Yeah. And lastly, you have this thing called superior colliculus, right? Okay, that's part of our, yes, it's part of our thalamus, right? So it guides movements of the eyes and head toward a newly detected object. Yeah. Okay, right, let's carry on. So these are the 3 kinds of layers, right? They're also named after the 3 kinds of ganglion cells, if you saw this thing, right? Because all these words are taken from Latin. So magno means big. I'm not so sure about parvo and conio, but they are much smaller, right? So the P and the K layers, they're highly involved in processing color, right? Similar to the P and K ganglion cells. M is for motion, right? Always remember, M is for motion. Right, so from the LGN to the occipital lobe, right? So the only thing, the most important thing that I want everybody to remember is this letter X, okay? So it's an X-shaped pathway, okay? Meaning that information from our left eye, right? Most of the info from the left eye gets passed to the right hemisphere, okay? Not all the information, but most of the information, right? And vice versa, most of the information from the right eye gets passed to the left hemisphere. Right, so now let's talk about these pathways, right? From eye to primary visual cortex all the way at the back of our brain. So our visual system has this cross-organization, or more academically speaking, we have this contralateral organization, right? So information from the left visual field is sent to the right hemisphere, contralateral side, right? And vice versa, right? Info from the right visual field sent to the left hemisphere, right? Just as what I said earlier. Okay, so our primary visual cortex, right, is also called the striate cortex, or the V1 area, okay, for short. Yeah. So it's right at the back. Okay, I would call it Brodmann area 17, right? It's really right at the back. All right, you can never get this thing wrong, right? We have around 200 million neurons. Okay, that's a lot, okay, per hemisphere, right? So 6 layers, like most layers of our cortex, 6 layers. So there's this very important concept called topographical mapping, right? Your textbook is called cortical mapping, right? But the concept is really the same, right? So this is how we map objects from different parts of our visual field onto different parts of the striate cortex. Yeah. So this is actually a good layman explanation now. So basically we're correlating, okay, or we're mapping, right, a location of neural activity with the position of an object in the visual field, right? So this is how all this information is organized. And then cortical regions responding to inputs from fovea, they're much larger. Okay, they're given priority, they're much larger than those responding to inputs from the periphery of the fovea. So if it's some highly detailed, colorful image, we do have much more cortical regions devoted to processing such information. Right? So peripheral vision, not so much. Okay, cortical region processing area, not so much. Okay, striate cortex, continue, right? So it's made up of simple cortical cells, very interesting, we'll talk about them, and complex cortical cells, right? Sensitive to movement for the complex ones. Simple ones is more like location, orientation, angles, right? Okay, so this one is similar to what we have in our tutorial. I don't think we have the time to watch it, okay? Okay, so let's just talk about complex cortical cells, right? So this is really from our textbook, okay? So let's imagine, okay, we have this bar of light moving in the preferred direction. So a complex cell will just fire as long as the light bar falls within its receptive field, right? That's the left panel. So you can see all these spikes, all these action potentials, right? We are having depolarization, so it just fire. Doesn't really matter where the light bar is, if it's in the middle, if it's to the right of the receptive field. Wow, I mean, we get the same pattern of AP, right? And then the right part is more interesting, right? So they respond to leftward movement of the light bar only, not to the right, okay? So only when the light bar is moving to the left, then we get to see the action potential. So meaning that, right, this specific complex cell we're looking at, it is selective for processing stimulus movement. Yeah. All right, so now we come to this very complicated slide, very hard to comprehend actually. I mean, it took me a long while, okay? The first time I taught this, it was very confusing, but now, okay, yeah. So this is like how, geometrically speaking, okay, we try to do this thing mathematically in a geometric fashion, how we organize all these neurons, okay, in our cortex, okay, we're talking about cortex, we're not talking about the retina, the cortex. So we had two kinds of visual cortical neurons, okay, or modules, all right. So one is what we call the ocular dominance columns, all right. Basically, they're kind of processing information from our left and right hemisphere. So you guys can see when I'm pointing at the L, at the R on the right panel, L stands for left hemisphere, R stands for right hemisphere. Okay, take note of that. And then we have this orientation column, which can kind of merge together to form hypercolumns. Wow, what a name, okay? Okay, so basically it's like responding to all these different orientation angles of very, very simple stimuli. Okay, like a light bar, for instance, right? If it's at the right angle, at an angle of say 180, 270, right? Along this column, right, we have all these neurons, all these cortical neurons responding to them. Wow. So a cortical module is just an assembly, right? So we have two ocular dominance columns, right? We have 16 blobs of this thing called cytochrome oxidase. It's an enzyme actually, okay? We need this thing to give us a sense of color, all right? So a kind of cylindrical shape, okay? And then we have two hypercolumns. Yeah, so it's pretty complicated structure. Yeah, and we need all these so-called columns, right, to give us a sense of orientation, movement, color, right? Even when we are like looking at this slide, okay, we are really depending on all this, hypercolumns. Yeah, right. Yeah. Okay, so this is an easy slide, right? Visual analysis beyond the striate cortex. So just take note. So in addition to the primary V1 area, we have this V2 to V5, okay, dorsal stream, the where stream, right? This is our occipital parietal pathway, and then our ventral stream is like our occipital temporal pathway, right? So dorsal is processing where the object is. That's why we need the parietal cortex. So this is what we need the temporal cortex for that, right? Memory of the object identity. So visual perception. So we have two theories. Okay, very quickly I want to go through them. So there's this thing called hierarchical theory. It's classical theory focused on bottom-up processing. Okay, and then we have a spatial frequency analysis theory, which is much more recent. I've read a lot of papers on this. Okay, by the way, so this involves a lot of mathematics. Actually, it's not so simple. Right? If you like advanced statistics, the second theory is for you. Right. So the problem, right, with this hierarchical model of vision is that it cannot really answer the question, how do our memory or knowledge influence our perception, So this bottom-up model, it just simply cannot explain why we perceive, a dog, right? Like on this slide, I'm sure that most of us can see that a dog, right? Its head is pointing to the ground, right? And then we have a tree here, right? Presumably with a shadow, right? That's all the black area, right? So we actually need some kind of prior knowledge. In other words, there needs to be some kind of top-down processing. Okay, this is what we learned in our Intro to Psychology textbook as well, right? So this is what this hierarchical model cannot explain. And so people thinking about, how about we get the spatial frequency analysis, right? It's more mathematical. Hopefully it's much better, right? Still, I mean, just to be honest, this thing does not really put the emphasis on top-down processing because it is a mathematical theory, right? So we're actually using all these visual spatial frequencies and contrasts to explain how we perceive the world, right? This is a non-intuitive, complicated way of looking at things. So it works by performing mathematical analysis of all this frequency and contrast of these gratings, okay, on this panel that you can see here. So frequency, the number of bars over time, and contrast, difference in color intensities, right? So this is just a very simple way of explaining this, right? If you're interested, you guys can look at papers on this theory. Okay, so now from sensation to perception, okay, let's talk about depth. Right, we'll talk more about this thing in our tutorials, okay? So just remember, there's some existence of this thing that we call the molecular cues and binocular cues, right? Binocular cues are just much more important, right? For us to see 3D depth. Right? Molecular cues, we'll go into detail in our tutorial, we have lots of it. So this is one way, right? That, this is how we see 3D virtual reality movies. So we have to wear on these Polaroids, okay? And what all these Polaroids are doing is that it's kind of filtering light of different wavelengths, right? And that really, it's kind of one is filtering in a horizontal direction, another one is in a vertical direction. And then that's really giving us this 3D view, this sense of depth. Yeah. So basically it's disparity in the wavelength of lights, okay? Wavelength of all these colored lights that we're receiving. Okay, visual perception, coding color, right? So one way of explaining this is this thing we call the trichromatic theory, okay, which is just influenced by the way we mix colors. All right, Young Helmholtz, very classical 19th century theory. Still apparent. It still works. It's not really wrong, right? So very importantly, this theory just advocates, it argues for, right? I mean, just like when we do a painting, yeah, different color outputs to engender the perception of different colors along the color spectrum. So this is like we're just doing it, right, with all the inputs, right, and outputs from our combs, and we're kind of integrating all these neural signals, right, to get different kinds of colors. Okay, let's just do this for a while. Okay, activity time, right? Even though I know that we know what we should be seeing, but just try. Let's just do it for a few seconds. Set the dot right at the center of the flag, and then after that, shift your gaze to the dot in a white space. What do you see? Right? Very hard. I mean, even I do this on my own screen. I mean, I can stare at this thing forever. You're supposed to see the USA flag, okay? And then literally a USA flag with all the right colors, right? Okay. So what it's arguing for, okay, this is based on this opponent process theory. Again, we see this word again, this adjective, antagonistic. So it's kind of arguing that we have these three antagonistic color channels. Okay, literally this is just copy and pasted from our textbook, right? So we have this red-green antagonism, this blue-yellow antagonism, and this black-white antagonism, right? So it's not antagonism between combs per se, right? But it's actually antagonism between the neural pathways, right? The processes for processing these different pairs of colors. So if we're kind of processing red, okay, our energy in terms of neural resources, for instance, right, will be devoted to processing red instead of green. So after, if you just stare at red for a very, very long time, okay, the pathway for processing the red color will be overused. You feel so tired, the pathway will be feeling so tired. And then after that, when you look at a blank space, right, what your brain is telling your eyes to do is just to, we're going to shift all this energy towards processing green, okay, because the green color is antagonistic to red. Okay, so on a white space, you will be seeing green, right, because the pathway, okay, all the neurons for processing red are just fatigued. Yeah, so that's, in a nutshell, that's how this theory works. Yeah, surprisingly, it does explain color blindness. Yeah, so this is what I said just now, right? So we have this, all right, this antagonistic red-green center around this structure again. So it's the same thing, right? So, but this time instead of bars of light, we're talking about colors, right? So the center responds maximally; it gets depolarized to the red color, and then the surround responds maximally to green only, right? And then we also have this antagonistic blue-yellow center surround organization for the K cells, right? So the centers respond maximally to blue, the surround respond maximally to yellow, right? So, and then what our author writes very vaguely, I still think it's very vague, right? It is agreed that both theories are correct, okay? Yeah, this is a consensus in the neuroscience view, right? But it's kind of believed that the opponent process theory is kind of more correct because it kind of stretches beyond the retina. So it's not just about the processing of colors, right, at the retina interneuronal level, right? It stretches all the way to our visual cortex, right, and other parts of our cerebral cortex. So I'll talk more about it in tutorial, right? Okay, so we have two more slides, right? So visual perception, okay, color deficiency, also known as color blindness, right? So a few kinds of chromacy, right? Chromacy means that you can see the color, right? So trichromacy is what's happening. Like everybody's normal, can see all photopigments, right? So it's only when you have dichromacy, monochromacy, and okay, anomalous trichromacy, I'm not so sure, okay? I think the last bit is that you should be color deficient, you shouldn't be caught as colorblind, right? But dichromacy and monochromacy, you literally, you are colorblind, right? Dichromacy is red-green color blindness, yeah, most often. Monochromacy is extremely rare, okay? You only have one functional cone, extremely rare. Okay, so this is just for everybody to take home now, okay? This is not examinable, but I think this is a must know, right? Just to make everybody more educated. So the most common color blindness is red-green blindness. Okay, I have to admit, I'm not so sure which one I have, but it's just one of them. I think it's protenopia, right? Yeah, red and green, sometimes they look the same, sometimes no, right? So I'm not too sure, okay? So because our tests of color blindness, are not really foolproof. I mean, they can be kind of faked, I'm not totally convinced, right? So, but just take note of all these different terms. And then blue-yellow blindness is a bit more rare, okay, rarer. Okay, so you cannot distinguish between blue and yellow. Okay, I will fix this, right? And we have this tritonopia. Not good, huh? So all these colors of short wavelengths look pretty much the same. No good, right? The last part is extremely rare. I haven't come across a person like this. Okay, so it's only in this case that you can effectively call a person colorblind, right? You cannot really see any colors. Everything is, shades of gray and white. Yeah, okay, that's monochromia. So the last part is just take note of color contrast. This is just a revision from intro to psychology. So color contrast and color constancy. So contrast, we can see, right, the different colors, right, side by side. When they're kind of illuminated, right, we can see the difference between the colors. So constancy is that colors look the same regardless of illumination, right? So we are making use of it for a lot of our optical illusions. So the wow basically looks the same, right? The color of the font looks the same, irrespective of the background color. Yeah, so that's the color constancy concept is being made use of in a lot of optical illusions, right? Okay, so that's all I have for today, right? So I want everybody to come attend to your tutorials, okay? I'll talk about next week, I'll talk about preparation for midterm.